will see post-transcriptional modification in eukaryotic. You know, eukaryotic cell type is a complex than prokaryotic. There is different modifications. There is different organelles like reticulum endoplasmic, Golgi apparatus. Even there is a nucleus. Again, that nucleus is bounded by nuclear membrane. So, in general, eukaryotic cell type is a complex than prokaryotic cell type. In prokaryotic, once replication begins, transcription continues. Then translation begins, or protein synthesis carried out. In eukaryotic, there is different modification. You will see these details. If you are not a member of Nigi Academy, subscribe the video. If you are already the member of Nigi Academy, like and share the video. Let us continue. When you say post-transcriptional modification, it is a matter of processing of RNA. You know, when certain gene is expressed, the end product will be RNA and the protein. Different types of RNA: ribosomal RNA, transfer RNA, messenger RNA, and others. So the end product is RNA or a protein. For protein synthesis, messenger RNA is very, very important. Messenger RNA carry coded sequence which code for amino acid. So here, once DNA is replicated, it starts to convert into RNA. That is what you call transcription. The sequence which is in the form of DNA languages changed into that of RNA. The RNA format immediately after transcription is pre-messenger RNA or hnRNA. It is not matured. It is not enough. So post-transcriptional processing of messenger RNA is necessary to produce mature form of RNA and to produce functional transfer RNA and ribosome from this primary transcript. Pre-messenger RNA should be processed in case of protein synthesis. The processing or post-transcriptional modifications are capping, polyadnylation, splicing, and in some cases, editing may occur. After this messenger RNA is produced, which is a matured form, it moves to the protein synthesizing machinery, which is ribosome. It is found freely in the cytoplasm. So protein synthesis is carried out here on the surface of ribosome. Before synthesis of protein, as I have said, post-transcriptional modifications are very, very important. Capping, polyadnylation, splicing, and editing. Let us see one by one. Capping is nothing. It is addition of a cap at the 5 end. Suppose there is a 5 and a 3 end. There is a 3 end. There is a 5 end. At the 5 end, there is addition of a cap, that is what you call 7-methyl-guanosine, to the 5 end. This 7-methyl-guanosine is then in 2 steps. The first step is guanosine triphosphate is attached at the 5' end of pre-messenger RNA. Pre-messenger RNA, this one. After that, actually, this is helped by enzyme transferase, guanyl transferase. After that, methylation of guanosine triphosphate is occurred by guanosine methyl transferase. Guanyl methyl transferase. So methylation is occurred here. Look at here. 5' end, 3-phosphate is here, nucleotide, phosphate, nucleotide is messenger RNA. Triphosphase convert this into 2-phosphate. After that, again it increases into the 3-phosphate. Then it adds methyl, 7-methyl guanosine could be added at this stage. That is what we call a capping. This methyl is added by enzyme methyl transferase. Transfer methyl group from S-adenyl methionine to the guanine ring. That is how capping is occur. This capping is very important. The importance of this capping is 1, for recognition. 2, for protection. You know, in the ribosome, there is different enzymes. There is a different enzymes that may degrade this messenger RNA. So this cap is very important for protection. For protection, let me write. Function of cap. Function 1, for protection. Protection. Protection actually. For protection. 2, for recognition. Recognition here. Yeah. So it has these two functions. 1, for protection. There is different enzymes that may degrade this messenger RNA that move to the ribosome. It protects different enzymes. 2, for recognition. You know, there are different transfer RNA. Transfer RNA here. Transfer RNA do have an arm, especially anticodon arm. That anticodon arm recognizes code information which is carried by messenger RNA. So this capping is very important for recognition. The other modification which is very important is polyadnylation. Actually, this polyadnylation is nothing but addition of the sequence of adenine at the 3 end. In the first stage of polyadnylation, primary transcript is digested by specific endonuclease recognizing sequence AAU AAA, which is polyadnylation signal. Actually, cutting occurs about 10 to 35 nucleotides below the sequence. So a sequence rich in GU or U located at about 50 nucleotides below is an additional determinant of cleavage site. There is a cleavage factors. You can see the detail of this in my another video. Cleavage and polyadnylation specificity factor, cleavage stimulatory factors. Here are cleavage factors. Cleavage factors cleave at that position. So polyadnylation polymerase at about 250 nucleotide, a residue in case of actually in case of yeast maybe 100 to 100 a residues, but in general 250. Thanks to this polyadnylation tail, the messenger molecule is recognized as it is own, not of one molecule. So just like a cap, this polyadnylation which is found at the tree end is important for protection and for recognition. Even it increases the effectiveness of translation. This is a pre-messenger RNA. Here is 10 to 35 of nucleotide AAA. Then cleavage may occur around the GU by cleavage factor. Then there will be addition of adenine, which may be up to 250, or in case of yeast 100. That is polyadnylation. The other very important modification is RNA splicing. RNA splicing is nothing but it is the removal of introns from primary transcript and the connection of exon. Introns is nothing, it is a junk region, it is a junk element. It codes for nothing. Exon is coding region. It codes for protein. So splicing must occur very precisely because even a single nucleotide shift can move reading frame and completely change a sequence of amino acids. Most introns begin with GU actually, from 5' and end with AG. Exonic and intronic splicing enhancers and exonic and intronic splicing silencers aid in recognition of exons actually. So here, look at here. This is a 5' this is the 3'. The betweens are exon and this is introns, this is exon, this is introns, this is introns, this is exon. Mind you here, for each fragment there is a polyadnylation actually at the 3' maybe. So here in splicing they remove introns. They connect exon. Polyadnylation is added, capping is added. After splicing, the mature form of messenger RNA is produced. That is how messenger RNA is formed. So this is a post-transcriptional modification, capping, polyadnylation, and splicing, which is very important. In some cases, RNA editing is very important. So after all these processes fulfilled, protein synthesis begin. If not, that pre-messenger RNA is not matured to translate or to produce amino acids. Thank you for watching this video. Subscribe, share and comment on this video. Thank you very much.